Hey, this week we're going to be doing genetics. Uh, Last week we learned about DNA and transcription and translation, and you learned about the genetic code, right? And we talked about mutations, and when we talked about mutations, we understood that if you switch the code letters, that you can end up with a protein that's not made properly. Well, that understanding goes into our understanding of this week's work. And I'm going to do my best to make my lectures very short because the SymBio lab is really, really good, but it's going to take you a long time to work through it. I just want to give you enough information so that when you do the SymBio lab, you'll feel a little more comfortable with tackling it. So we're doing genetics. Oh, that was the other thing I was going to say. Last week we did DNA. This week we do genetics. Next week, or the next lab week, we're going to be doing evolution. And you need to understand that week and last week and this week if you're going to understand evolution. So let me just remind you about genes and alleles, okay? So a gene is a short segment in the DNA. DNA on one of your chromosomes, millions of base pairs long. A gene would be maybe a couple thousand base pairs long. And that is the coded instructions for how to build a protein. Now, all of us have genes for a protein that makes eye pigment or a protein that makes skin pigment, but not all of us have the same version of that gene. When there are instructions for making a protein, but the instructions are slightly different, so the protein ends up being slightly different, that is called an allele. So I've got an allele for eye color, but mine's broken. My instructions make a broken protein. That's my allele. Your allele, if you've got brown eyes, your allele makes a perfectly functioning protein, right? So we both have got a gene for eye color. It's just that we have different alleles. And so when people are saying, oh, I've got a gene for curly hair, no, you've got the curly hair allele at that gene locus. They call it a locus, L-O-C-U-S, locus. Okay? So we're going to be thinking about alleles. Ultimately, evolution is the change in the frequency of alleles over time. Sounds much more boring than what everyone argues about, but it actually is the real science of what's going on with this thing we call evolution. So we all have genes for different things, like we all have genes for arms, but your allele might make your arms longer and my allele might make my arms shorter, okay? But we all have the same genes, we just have different alleles of those genes, okay? Now, some alleles, they make proteins that don't work right at all, and others have proteins that work a little too extra good. Those subtleties are going to be part of your SymBio lab. So I've told you that proteins, many of them are like robots. Here are two versions of Voltron, the toy. They're both Voltron, so these would be the same gene, the same toy, but two different versions of it, two different alleles. This one over here can fly. This one over here cannot fly, but has got more arms, whatever that's good for, all righty? So everyone's got a gene for all these human characteristics. We just have different versions of it. We have different alleles. Okay? In your SymBio labs, they use the example of coat color in pigs. And I don't remember all of the genetic mutations, but black pigs, they've got a pigment making protein that makes black pigment all the time. And so those pigs are black. Brown pigs have got a version of that protein. So they've got an allele that creates a protein that it turns on and off. So they are not black, they're brown. There are also red pigs and spotted pigs, and so different alleles. Now, another thing I want you to know before you get going is about Punnett squares. There's nothing in the SymBio activity that actually teaches you how to use a Punnett square. A Punnett square is like tic-tac-toe or something like that. It is not complicated, but man, it really allows you to solve a lot of these problems. And I found it very useful to use Punnett squares to answer many of the questions that they were asking me in the sim bio activity. So I'm going to work through a couple with you. So you need to know the term homozygous and heterozygous and dominant and recessive, okay? So Shirley is homozygous for blue eyes. If she's homozygous for blue eyes, she's got 2 of that allele. Clyde is heterozygous for brown eyes. That means he's got, in this case, he has 1 allele that says make brown eyes and 1 allele that says make blue eyes. So he's heterozygous. Now, it also tells us that brown eyes is the dominant trait. So in the case of this eye color hypothesis, eye color in humans is not exactly this simple, okay? But in this case, this means that Clyde has got 1 allele on 1 of his chromosomes that's broken, can't make brown pigment. But he has another allele, and that other allele allows his cells to go ahead and make brown pigment, so he makes brown eyes. Now, not in the symbio activity, but in this activity where we're doing dominant and recessive, we very often will write the dominant allele as a capital letter, and we will write the recessive allele as a lowercase letter. So Clyde is heterozygous. We're going to make him across the top. If he is heterozygous, then his alleles are capital B, lowercase b. But when he makes sperm, half of his sperm get this allele and half of his sperm get that allele. Okay, so half of his sperm, brown-eyed allele, half of his sperm, blue-eyed allele. Shirley's going to be over here, and she's lowercase b, so when she makes eggs, all of her eggs have got a lowercase b because she's homozygous for that trait. Great. So now what we're going to do is we're just going to fill in the Punnett square. So if this sperm bumps into this egg, this will be the genotype for that particular offspring. And again, we'll do this one with this one, right? Capital B, lowercase b, right? Now, if this egg bumps into this sperm, we get 2 lowercase b's, and ditto here. Okay? So let me get out my eraser and erase some things to make things a little easier. Okay, so now we know that out of all of the offspring that these 2 can have, these are the 4 different ways that egg and sperm can combine. And what we are looking at is the genotype of the children. So when I look at this and it says, what are the possible genotypes of their children? Their children could either be heterozygous for the brown-eyed trait, or they could be homozygous for the blue-eyed trait. Those are the only 2 options for these particular parents. Okay. Now, when we ask about phenotype, we don't mean these genes, these alleles. We mean, what will their kids look like, right? Well, when their kids have got heterozygous, they're just like their dad, and they've got brown eyes. So half there, and these guys will have blue eyes. Now, what are the odds? What are the chances that any kid that they have will have the dominant phenotype? The dominant phenotype is brown eyes, and the odds are 50-50, or 1-to-1. Okay, let's do another one. Here we go. Mandy has a round face and Stan has a square face. Mandy's father had a square face. A round face is the dominant trait. All right, now make a Punnett square. So this is more complicated than the last one. a little more complicated because they didn't tell you whether Mandy was homozygous or heterozygous. But go ahead and pause me and try and work it out on your own because I'm about to do this one. Okay? Now, Mandy has a round face. So I'm going to use the letter R for face shape. And they tell us that a round face is dominant. So capital R will be a round face and lowercase r will be a square face. And I know that Stan has a square face. And since that's the recessive trait, that means Stan is lowercase r. So when he makes sperm, half of his sperm will get a lowercase r, and so will the other half. Yes, because he has got the recessive trait, so he is only carrying the recessive allele, which in this example is evidently square. Right? Now, what about Mandy? We're not sure about Mandy. We do know she has a round face, though, so we know Mandy has got one uppercase R, but they didn't tell us what the other allele is. However, we know that Mandy's dad had a square face, so Mandy's dad was like this. Mandy's dad only made sperm that carried a lowercase r, so Mandy has got one dominant allele and one recessive allele. So she will make eggs that are capital R and eggs that are lowercase r. So if we work this Punnett square, we will get capital R, lowercase r, and then capital R, lowercase r, and then we'll get lowercase r, lowercase r, lowercase r, lowercase r. Ready? So the genotypes of their children, half will be heterozygous for a round face, half will be homozygous for a square face. And what will their phenotypes be? Half will have a round face and half will have a square face. All right, that's how you do Punnett squares. I hope that's helpful. And I'm posting up a Khan Academy link for those of you who want more help with Punnett squares.